Kia ora, and thanks for joining us today. My name's Christine Harper, and I'm the Stakeholder Relationship Manager at Manaaki Whenua. Sharing the great work from our research is one of the most exciting parts of my job. And I know that you might have quite a few questions. Please put those into the chat box as soon as you think of them. We will do our best to answer all of the questions if we can, but those that we don't get to in the session today, we will collate and upload responses to our website along with the recording of the webinar. This is the final of a series of webinars about our work and the results from the Kiwi Rescue research program, which had many partners and collaborators working together over a 5-year period. Danielle Middleton is our presenter today. She is a molecular ecologist at Manaaki Whenua and has led the molecular component of the wider Kiwi Rescue program for approximately 3 years. Danielle's research is varied and includes all aspects of molecular ecology, from genetics to microbiology. She's found working on the Kiwi Rescue program particularly rewarding due to the close collaborations with end users, and is really proud of the tangible, practical tools the team have produced through the program. Over to you now, Danielle.
Yeah, thank you, Christine. I'm hoping that everyone can hear me okay and I'm not skipping too much, as I really struggled with hearing Christine and Tiffany there. So I believe I may be having some issues with my audio. But nonetheless, let's launch into this. So yes. Oh. Christine, were you saying something there?
Just keep going, it sounds good.
Okay, great. Yeah, so as Christine was saying, this is part of the wider Kiwi Rescue program. But today we're going to be talking about the molecular component of the Kiwi Rescue program. You have heard from a number of my colleagues already, most recently from Paul Schofield last week, who talked about the Kiwi taxonomy. But today's presentation focuses on some tools that we have developed, which we hope you might be able to pick up and use to help Kiwi survive. So first of all, a little bit of the usual disclaimer, if you like, that whilst I am the person fronting this presentation today, there are numerous people that have been working away in the background and have contributed at least as much, if not more than myself, to many components of this research. So I'd like to give a big shout out to Talia, Julia and Hester from Manaaki Whenua, but also to Hugh Robertson and Rogan Colburn from the Department of Conservation. I'm not sure if Hugh and Rogan are able to make it onto the call today, but without their vast expertise and just general willingness to help, we certainly would not have been able to get as far through this research as we have. So as we've said, this is part of the large Kiwi Rescue Endeavour program. And what I've got here is a sentence that was taken from the public release when the funding was first granted. And the sentence says, working with DOC and Kiwis for Kiwi, we will clarify how many Kiwi species there are, develop new DNA-based techniques to estimate Kiwi population size from feathers and scats, and identify predatory dogs, stoats, and ferrets at the individual, not just species level. So today, I'm going to talk to you about the bits in red there. Essentially, how can we use DNA to better estimate how many Kiwi there actually are out there, and also to understand those predator-prey dynamics. So what do we know about Kiwi at the moment? Well, our knowledge of both how many Kiwi there actually are in the wild and the rate at which they're declining is a little fuzzy, shall we say. We know that, or we estimate that unmanaged populations of Kiwi are declining at a rate of around about 2% per annum. But that is an estimate because we simply don't have very robust estimates for how many kiwi there actually are out there in the environment. And those estimates, or the accuracy of those estimates, vary greatly across the taxa. So across all taxa, we estimate that about a quarter are under some sort of management. And by that, I mean either they're really highly actively managed, breeding programs, extensive predator control, all the way through to they have a little bit of predator control around the site. But a whopping sort of 75%, so 3/4 of them, are actually unmanaged. And as I say, that varies across the taxa from things like our Rakiura tokueka, which is quite an abundant population, so we have about 2% under active management, all the way through to our little spots and our rowi, which are actually quite threatened populations, where we have, almost 100% or 100% under active management. So one of the things that we wanted to achieve through the Kiwi Rescue program was to, one, get a better understanding of how many kiwi there are actually out there, and two, try to minimize that decline by trying to restrict the amount of predation that was happening. So I want to say that this isn't going to be your normal sort of scientific talk with graphs and PCOA plots. More, this is a presentation about the tools that we have developed. So the R&D has been done under the Kiwi Rescue program, but these tools and techniques are now available for all of you, the practitioners, to use through Ecogene. So Ecogene is the business unit of Manaaki Whenua, and it does DNA-based diagnostics for wildlife. And all of the tools and techniques that I'll be presenting to you today that we developed under the Kiwi Rescue program are now available as a fee for service through Ecogene. So as you'd expect, because we know it's a problem for all of our native and endemic flora and fauna, kiwi are under threat by mammalian predators. Dogs are a major problem for them, but they also fall prey to all sorts of other mammalian predators like ferrets, stoats, cats, even pigs and rats. What this graph here is showing you is the number of kiwi predations that were sent to Ecogene for identification over the last few years. And through the Kiwi Rescue program, one of the things that we wanted to do was to minimize the number of cases that appear in this sort of grey box. The number of cases where we were unable to identify a predator. So what we're talking about here is forensic DNA sampling. And just like human forensics, we're dealing with very, very, very small amounts of DNA and very highly degraded DNA often. It can sometimes be days, weeks before we actually get a sample, so a dead kiwi, through to do this DNA analysis on, by which time we're dealing with very highly degraded DNA. So we were hoping to design a range of tests that would be more sensitive and allow us to minimize that grey box. Now, some of you will be looking here at 2020 and thinking, well, Danielle, you failed miserably at that. I'd like to debate that. Of course I would. What we're seeing there in 2020 is, well, 2020 basically. Most of you know and remember that you were in lockdowns, in and out of lockdowns. It was really hard for you to get out and check on your populations. Therefore, there was often a delay between the kiwi actually being killed and us receiving the sample. So we were dealing with highly degraded forensic samples, and obviously that affected our ability to identify the predator involved. You'll also notice there that there were less predations sent to Ecogene for sampling in 2020. I don't think that means that there were less predations happening in 2020. Sadly, I don't think the stoats and the dogs got the memo that they were supposed to stay home and be kind. I do think they were still out there killing our kiwi. But again, all of you lovely people, you weren't able to get out in the same numbers that you were in 2019. And also, we know that 2020 came with a huge financial blow. So there may not have been the money available for you to send samples to Ecogene for testing. But one thing that I think we can all agree on from this graph is that dogs, which are here in these sort of dirty orange bars, are a major problem for kiwi. So let's start with them. We wanted to, as I say, not only minimize the number of cases where we couldn't identify if it was a dog, but we also wanted to be able to identify the individual dog that was involved in this kiwi predation. And so to begin with, we canvassed the scientific community to see what tools were already out there. And we actually came across this tool known as Dogfiler, which was developed in the States for dog attacks, but more importantly, actually, for those illegal dog fighting rings. And we introduced it into New Zealand and we adapted it for use in a Kiwi forensic setting. But more importantly, actually, for a conservation forensic setting. That tool that is now adapted and fully in use by Ecogene has now been used in a number of civil prosecutions under the Dog Control Act. But most excitingly, well, sort of excitingly, not excitingly for the Kiwi, but one good thing that came out of 2020, I guess, was that we saw 2 criminal cases tried successfully under the Wildlife Act. And for those of you who are aware of forensics, either humans or animals, you'll understand that having this all-important case law means that Dogfiler as a tool has a much better standing and will be able to be utilised in forensic conservation cases more frequently in the future. So I should say that here I'm talking about the use of Dogfiler on kiwi, but Ecogene has done dog versus postman, dog versus sheep, dog versus penguin. So it's certainly a tool that you can use when you're researching your other iconic bird species or species that are likely to fall foul to dog. A couple of weaknesses with this tool: dogs often attack in packs, and as I say, when you're dealing with very small amounts of DNA anyway, when you end up with small amounts of DNA from lots of different individuals, that really blurs your signal a lot and can affect whether you can successfully identify an individual. I know this is something that the team at Ecogene are keen to continue to develop beyond the life of the Kiwi Rescue program. So watch this space. The other thing is that just because you find dog DNA on a kiwi, it doesn't necessarily mean that that dog killed that kiwi. One way that we manage to, or that we increase our sensitivity and specificity around this is by swabbing the actual bite wounds and by taking multiple swabs from multiple bite wounds across different sites of the body, particularly around the areas of the neck and things where the dog's going to grab on and shake. And if we get the same signal from multiple bite wounds, then that increases our confidence that we've actually identified Max the dog as the killer, as opposed to just came up and sniffed it or licked it post-mortem. But sadly for kiwi, dogs are not the only issue they have. So again, we went out, we canvassed the scientific community for tools that we could use for mustelids. And we found this ferretplex. Ferretplex is great at identifying individual ferrets again, but only when we have pretty good quality DNA. So this tool was developed for ferrets as a TB vector, and it uses a technology known as microsatellites. So microsatellites are short, repetitive regions within your DNA, and they're quite simplistic in that they're often repetitive nucleotides, AT, 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 or GTC, GTC, GTC. But they are still blocks of DNA, and so they don't occur that frequently within your genome. And therefore, when you're dealing with highly fragmented DNA, such as forensic DNA, it doesn't always give the best signal. So for stoats, we wanted to develop something that would be much more sensitive. And to do this, we looked at a tool known as single nucleotide polymorphisms, or SNPs. As the name suggests, this is a variation in a single nucleotide. And because it's a variation in a single nucleotide or a single site, it occurs thousands of times throughout your genome. So looking at stoats, we identified more than 50,000 of these variable sites in the stoat genome. And we did that by looking at 94 stoats across 15 populations. And from that, more than 50,000, we narrowed it down to just 200 that were the best at identifying individual, sex, lineage, and ancestry. You might be thinking, well, if it can identify individual, why are we worrying about sex, lineage, and ancestry? And again, it comes back to everything being so degraded. So if we cannot, because the DNA is too badly degraded, identify individual, then at least we might be able to provide you with some information, such as whether it was a male or female stoat, or which family group it came from. We see this tool being particularly useful when you get island incursions of stoats or ferrets, and you want to be able to identify whether you've caught the right stoat or whether you have more than one. Here's another example of how it can be used to look at the sort of regional diversity of stoats throughout New Zealand. From that SNP panel, hopefully here you can see that the North Island stoats are more closely related to each other than the South Island stoats, and we've got Wellington smack in the middle, as we'd kind of expect. Hamner Springs is a bit of an anomaly there, and I believe Andrew Veal is hoping to investigate that a little further. So far, I've presented you with tools that you can use when you're faced with a dead kiwi. So you actually have the kiwi in front of you and you want to know who killed it. But another tool we wanted to be able to provide you with was the ability to know whether the stoats and the ferrets in your region or on your mainland island were eating kiwi. And so we wanted to design a tool that could identify Kiwi DNA in fecal samples. And we did that using a probe-based DNA tool. For those of you who don't come from a molecular background, the best way to think about that is literally as a probe. So you can imagine that in feces, there's a huge amount of DNA, bacterial, host, everything they've eaten. And you're going to take a probe and you're just going to pull out the one piece of DNA that belonged to the kiwi, and you're going to amplify that. So using this probe-based tool, we were able to identify Kiwi DNA down to the tens of femtograms. For those of you like myself, where high school was quite some time ago, 1 femtogram is 10 to the negative 15 grams. So that's a huge amount of, well, that's a very, 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 very small, rather, amount of DNA that we're able to identify in this poo and soil samples. Now that was done in vitro by doing spiking studies. So we literally took the poo and we just added less and less Kiwi DNA to it. What that didn't tell us, though, is what happens when the kiwi, and therefore the kiwi's DNA, passes through the predator's gastrointestinal tract and is exposed to all of those acids and things. So to do that, we looked at doing a feeding trial in which we fed 3 captive stoats kiwi meat. and they were offered some leg and liver and heart, and they were given that for 48 hours. And we observed all 3 stoats numbing up the leg muscle meat. They thought that was delicious, less keen on the offal that was barely touched. And that sort of corresponds to what we'd see in the wild, but the most important thing is that we saw them eat the kiwi meat. And then we collected the scats from those stoats, and we were able to identify Kiwi DNA in those scats for more than 5 days. So we actually stopped looking after 6 days, which turned out to be a little bit of a mistake. We thought that the DNA would have passed by then, but we were still getting Kiwi DNA signal at 6 days. So that's really exciting, and it means that you could potentially grab some scat from the wild, send it to us, and we would be able to tell you whether your stoats were eating kiwi. And we believe that this would work just as well in any other kind of predator feces. What we can tell you from that is that we can identify individual predator from the poo. So we could tell you that Max, the stoat, has been eating kiwi, but we could not tell you that Max, the stoat, has been eating Hillary, the kiwi. By the time the DNA has gone through the gastrointestinal tract, it's so highly fragmented that even using SNPs, we're not really going to be able to identify individual kiwis at that point. Okay, so just to finish up then, I want to move away from predators to estimating effective population sizes and giving you information about the number of kiwis that you have in an environment. So we wanted to develop a tool, a molecular tool using feathers that would allow us to estimate population trajectories and estimate effective population size. So my predecessors went some way to achieving this by developing the actual molecular technique. We've since tested this technique using samples that were very kindly supplied to us by Hugh. And we find that using feathers, we can estimate effective population size, and that matches what's known based on census population size, which has been calculated by dog surveys. So this molecular tool is very, very sensitive and should work very, very well, provided you can get feathers in enough quantity to be able to estimate effective population size. Can you collect feathers and scat in the field in sufficient quantity and quality to enable estimation of population size? Well, as a Kiwi, talking about Kiwi, here's my Kiwi answer. No, not using the tools that we used. You will have all heard of the proverbial needle in a haystack. I'm going to start a new one, which is looking for a Kiwi feather on the forest floor. So look, it's just not possible using the tools that we used, which was like walking transects, to find enough feathers to be able to estimate effective population size. Certainly this isn't a write-off. It can be done. It would probably need trained Kiwi dogs to look for the feathers and scat, and you would need to weigh that up against using the dogs for that versus using the dogs for other types of surveys. You could also use your volunteer armies to this end, but again, you need to weigh that cost-benefit analysis up. There is one other thing that has arisen from this though, and that's that probe that we talked about where we found Kiwi DNA in scat. We can also find Kiwi DNA in soil. Again, because it's so highly degraded in the soil, we can't identify to the individual level, so we can't estimate effective population size. But we believe that we would be able to provide other estimates of population parameters that may be useful. So watch this space for that one. It's something that we are going to continue to explore into the future. But essentially, we can use feathers to estimate effective population size if we can get them in high enough numbers. So I see it's coming up to 11 o'clock. So I want to just use the last minute to say thank you so much to the far too numerous people who contributed to this research, who spent many hours trying to collect feather samples, who sent us stoats and ferrets and predated kiwis, who commiserated with us and celebrated with us. Yeah, we just want to say a massive thank you to you.
Kia ora, and thank you, Danielle. That's really great. We have not got a lot of questions, but that's good because we haven't got a lot of time either. I do have one though. Your histogram of ecogene predator IDs had fewer stoats than Tim might have expected. Did that data include kiwi chick samples from Shy Lake over the last few years?
Yes, it did, but not the 2020 samples because those are yet to be processed.
Okay, thank you. So look, we don't have any further questions. We will wrap it up here and give everyone back 2 minutes of their precious time. I'd like to say a big thank you to Danielle and to all the presenters who have shared their experiences through this 5-year research program with us through this. While the research comes to an end, obviously the usefulness of the research certainly doesn't come to an end, and it's been good to be able to leave this series on the note around how we have practical tools to help everyone to improve the fate of our kiwi populations. So with that, I'll say goodbye and we'll be in touch about our next webinar next month. Thanks. Bye.